My thanks for the invitation to be here today and share our work with you. Trophoblast retrieval and isolation from the cervix, we often use the acronym TRIC to refer to it. It's an alternative approach for doing non-invasive prenatal testing, and we've been developing it for the last few years. It begins with a cervical sample, which is basically like taking a pap smear. So we use a cytobrush. It's collected in the standard collection kits that are available in most clinics. And then Brian Kilburn, who works with me, has done a lot of the development of this with me. He's been the person to get all this to work. And basically what we do is we select trophoblast cells which express the antigen HLA-G when they're extravillous trophoblast cells. And so antibody to HLA-G is coupled to magnetic nanoparticles mixed with the cells, and so the red would indicate the trophoblast cells, the blue, the maternal background cells. They magnetize the trophoblast cells, which can then be isolated by magnetizing a test tube and washing it several times to remove the maternal cells, and what you're left with is a fairly efficient and highly purified sample of trophoblast cells. When we analyze these samples, we find that here we're staining for HCG, a trophoblast marker, in green, and you can see most of the cells that are co-stained with DAPI light up with the HCG, whereas the excluded maternal cells do not. And then this is a control with IgG instead of the antibody. We can also, in the case of pregnancies that have a male fetus, do FISH for the X and Y chromosome, and we'll see most of the cells are staining for both the Y chromosome and the X chromosome. So what we believe is happening is that extravillous trophoblast cells are migrating from the placenta into the uterine cavity and then finding their way to the cervix where they can be collected in this pap smear procedure. These are interesting cells because the extravillous trophoblasts are the cells that are disrupted or thought to be disrupted in diseases related to the placenta or poor placentation, such as preeclampsia or IUGR. And of course, these cells would reflect the fetal genome much like a CVS sample. It's coming from the placenta. So the extravillous trophoblast cells migrate into the uterine cavity carried by uterine secretions, and we use an FDA-approved cytobrush for collection. And it would be something like this, where the cytobrush is inserted into the external os, rotated a few times and removed to collect the sample, well away from the conceptus. We believe that this is a safe approach. Pap smears are recommended during pregnancy when needed. And in our experience, we've done well over 2,000 of these samples now from ongoing pregnancies. Here we analyzed about 553 samples, and we found that the pregnancy loss rate was quite low. In the first trimester, we saw a rate of about 1.5%, which is on the low side, and in the second trimester, about 2%. Most of these losses were only 2 were within 2 weeks of sampling. Most of them occurred 2 to 8 weeks or later after the sampling was done. And there's also a fair amount of literature suggesting that it's safe to do this. So it can be considered a safe approach. Anthony Amudia worked with us early in these studies. He did a review of the literature, which actually goes back to the early 70s. Various ways people have collected trophoblast cells using either uterine lavage, which is not considered non-invasive, of course, but they've also done lavage of the cervix, the cytobrush method as we have had, and other methods. They've identified the trophoblast cells there either by using fish to look for males in half the samples, or using this anti-HLA-G antibody, which is fairly specific for at least extravillous trophoblast cells. So how do these cells come into the cervix? So we've worked with Gerrit Moser and Berthold Huppertz at the University of Graz. They have a collection of archived tissues, and they've been studying the invasion of uterine glands by trophoblasts. And so we worked with them to look specifically at routes that might lead into the uterine cavity. So here you see a section at the junction between the decidua basalis and the decidua capsularis. So it's uterine cavity to the upper portion and intervillous space below. If we expand that red area, what you can see is a uterine gland, and it's secreting into the uterine cavity here. Adjacent is another example of a uterine gland. They're stained blue with antibody to cytokeratin 7, and then the brown stain is against HLA-G to pick up the invading extravillous trophoblast. And you can see one of these extravillous trophoblast cells breaching the epithelium and entering the uterine gland. And over here is another one that is already in the gland. And you can also see some extravating from the surface of the uterus at the uterine epithelium and moving into the cavity. Some more examples here. A couple of areas blown up where the uterine cavity has come in next to the decidua capsularis towards the basalis, and you can see some examples of brown staining extravillous trophoblast cells that are moving into the uterine cavity or have already entered it, down here in some secretions. So this is the route that we hypothesize leads to these cells entering the uterine cavity and presumably being carried to the cervix. And this just kind of illustrates the cells coming from the margins of the placenta as it's developing and growing. And so those that enter uterine glands at the edge that empty into the uterine cavity would be carried there. And just to illustrate a little further, they could either come from a gland, as you see above, or possibly directly where there are villi still in the capsularis region for a while. So this compares, if we take a look at the invasive procedures, amniocentesis, which is done beginning around 15 weeks in the second trimester, or CVS, which can be done as early as 10 weeks. They're invasive procedures. They have a certain amount of risk, but they directly sample fetal material. This enables full genetic analysis. We're not dealing with fragments of degrading DNA with a fast turnover rate as you would have in NIPT, and you have much lower maternal background to deal with. Of course, you have the risk with those, and they're more complex procedures. And then NIPT, of course, we've talked about quite a bit. It's safe, there's no risk, and it's not a fairly complicated procedure to get the blood sample. It becomes more complicated in the genetic analysis, of course. As we've heard today, there's a lot of effort to be able to get more and more information, and we're making great progress in that area. Of course, it's, one thing, it's not able to be used much earlier than about 10 weeks also, or 9 weeks. So again, after the fetal heartbeat is formed. Now, some of the earlier analysis of our work was done by Ronnie Fritz here. He looked at 224 of our early samples. The average purity based on HCG was 96%, so we get a fairly high rate. The yields were quite good, 730 cells. So we get hundreds of cells. It's more than you would get with pre-implantation diagnosis, of course. And it's probably less than you get with the invasive procedures. But with hundreds of cells, you can more directly do genetic analysis. It has the other advantage that it can be done quite early. We get as many cells at 5 weeks of pregnancy, so 1 week after the missed period, as we do out to 20 weeks. After 20 weeks, there's a bit of a drop-off, but it's fairly flat, as you can see. It's not affected by obesity, which is a problem with NIPT because you get more background cell-free DNA. So again, the comparisons are with the invasive methods. You have the risk issues. With NIPT, you have a very low fetal fraction. With TRIC, we can reach up towards 100% for a fetal fraction. So again, it makes DNA analysis quite straightforward. It's also going to be potentially more reliable for looking at monogenic disorders and doing whole genome sequencing, those types of methods. It has the added advantage that it also can be used to look at indicators of pregnancy disorder, since these are the extravillous trophoblast cells, which play such an important role in early placentation. Today I won't have time to really talk about that work, but we've been able to see that there's some potential there. So leveraging TRIC, we can look at it in terms of doing prenatal genetics. It has the limitations of CVS. Again, we're looking at placenta, so mosaicism could be a potential problem, but we should be able to look at single gene disorders, aneuploidies, possibly even doing whole genome sequencing, DNA methylation, those sorts of approaches. Additionally, we have the ability to look at biomarker discovery for obstetrical disorders through transcriptomics, proteomics, and other discovery approaches. And then it also has some use in terms of placentology, just studying how the early placenta develops, because we can get cells from the early placenta as it's developing and without disrupting the pregnancy, and then find out what happened. So if you could culture these cells, you could potentially establish cell lines that are associated with different obstetrical disorders. You could potentially dedifferentiate or transdifferentiate them into other trophoblast phenotypes. So it has a lot of potential there. So my collaborator, Sasha Drulo, joined us at Wayne State about the time we were first working out the details of TRIC. He has a great background in placental disorders, and he's a very good molecular biologist. I am less so, so it was a nice opportunity for us to team up. And we had a nagging problem at this point in the project. So the good thing is that there's approximately 600 picograms of DNA available from just 100 cells, which is enough to do sequencing. We have the complete genome present, and there are all these possibilities. When we've done TRIC followed by FISH, as you can see here again, where it's a male, we can see almost every cell has a Y chromosome, so it has very high purity. But the problem was when we took these samples and we did sequencing for SNPs to see how different it was from the maternal fingerprint, as you will, we got something like this. So when we compared the maternal versus the fetal, and we're just plotting SNPs here, so they're either, it's the number of reads for different markers, and you could see that they're identical between the maternal and the fetal. that there's no difference here. Whereas when we compared the fetal that we got by TRIC to the placenta from the same pregnancy, now we see differences. So it appeared we had all maternal DNA. So the next challenge that Dr. Drullo and I worked together on was how to get that DNA pure. What was the problem? And we knew from doing FISH that we just saw 2 copies of each gene when we looked at different genes. So it didn't appear that during FISH we were seeing the maternal contamination, if you will. So that suggested that when we do FISH and we isolate, we degrade the cytoplasm so we end up with just the nuclei, that cytoplasm was the source of the contamination. So we basically mimicked that procedure. As shown here, we fixed the cells to a slide, treated them with pepsin to remove the cytoplasmic components so we were left with just nuclei. So basically isolated nuclei. And then we isolated DNA from that. And so then we could go on from there. So this kind of illustrates it. And so we did a procedure where we fingerprinted the DNA similar to what I just showed you earlier, looking at both SNPs and STRs through an Illumina product called Veriseg, which looks at approximately 150 SNPs and STRs. And they're located throughout the genome. And what we found here, you can see with the STR analysis, we have the maternal DNA, the DNA obtained by TRIC, and matched placenta DNA. And in some cases, it was non-informative. Other cases, it was informative, as you see in the 2 lower examples here. And you could see the maternal is different than what we got with TRIC, and TRIC is identical to the placenta. And the same thing was true when we looked at SNPs. Going across here, maternal versus fetal. The blue maternal is lined up according to read depth, and then plotted over that are the read depths for the fetal, and you can see the differences that we're picking up here. Whereas when we compare it to placenta, the SNPs line up exactly. So it looked as though we were getting success in getting the pure DNA from the fetus. And then this was some examples. Lena Kadam and Reza Kohan-Gadir were 2 people that worked on this work and are still working with us on the project. And you can see the gestational ages of the various samples shown here. These are 20 samples. A lot of them were early first trimester. The trophoblast purity based on HCG staining of a portion of the sample was in the 80s to 90s. The fetal genders were evenly distributed, and the fetal fractions determined from that forensic analysis were anywhere from about, I think the lowest was 82% up to what essentially 100%. So this is a rigorous method for interrogating the fetal DNA. Fetal haplotypes matched the placenta and were different than the maternal DNA. So from this, we've gone on and tried to look at it as a way to look at pathologies. And the one we started with was sickle cell disease because our hospital is in an inner city and we have a large African American population. Many of them have are carriers of sickle cell disease. So this was a single nucleotide change, as you know. So it was a nice way to start at looking at our ability to haplotype. So we determined the read depth for each allele. As we went along, so we're doing targeted sequencing. So we have PCR amplification around sites of interest. And what we did is multiple PCRs across the hemoglobin B gene. And so we got all 3 exomes here fairly well covered, shown in the light blue. And then from those read depths, we can make determination. And so we not only get the sickle cell site, but we get the other sickle cell-like sites and other hemoglobinopathies, such as beta thalassemia, can be determined from this. You can see in 30 samples here, this is preliminary data. We have median coverage depth of about 3,000-fold. The number of good calls was about 80%, and the median coverage was 12 to 19,000 over this. And we were able to get 64 hemoglobin B loci detected per sample. And then some more on the performance. So we use clinical records to identify either healthy or carriers that were identified. We confirm by sequencing corresponding placentas in 12 cases. And you can see down here sensitivity, which is not that meaningful at these low numbers, but this is our initial attempt, and this is ongoing, and we're planning to expand this data. So we're also interested in detection of aneuploidy. We've been working with Susan Klugman at Albert Einstein. We've gotten patients through them, as well as some of our own patients we've just randomly selected as potentially controls. We've looked at the fetal and maternal DNA isolated by TRIC. We use Forensic to determine a fetal fraction. We used whole genome amplification followed by Illumina's Veriseg platform to analyze chromosome number. And we've also done some FISH for confirmatory. Our test results shown here, 18 samples were basically randomly chosen. You can see the fetal fractions. They were all disomic by Veriseg, whereas 2 samples we obtained from Einstein that were suspected of trisomy 21, we got high purity, high fetal fractions, and we were able to confirm that by Veriseg that they were trisomy 21 in each case. This is the example from the one of the disomic samples here. You can see in the fetus obtained by TRIC, it's a normal male. It has 1 copy of X and 1 copy of Y, and it's otherwise disomic, as is the placenta, and then the mother is female disomic. Whereas this sample from Einstein below, the fetus, you can see a blip there at 21, and also in the placenta. This was a female fetus. And then if we look at higher, kind of blow up the area around there, you can see the increase for trisomy 21. So you can get a fairly clear picture of the aneuploidy in these samples. Okay, and then this was just some FISH analysis of that sample, showing down below here, you can see some cells. The lower one, you can see the 3 labelings of 21, and it's normal for X, Y, and 18. And then some more examples. The red is the 21, which you see 3 labels for, and then excluded cells, which would be maternal, are all disomic. So we've also done, made an attempt at exomic sequencing. These were 5 samples that were all male fetuses. And you can see here that in the TRIC samples, they have both the X and the Y showing up, whereas in the maternal sample, they're all X. And then down below, some references. And then this shows actually a little nicer. You can see the pairs of maternal and fetal DNA in orange and the amount of labeling we got, which was fairly even across the exome. And then blowing up this last part where you can see X and Y, and you can see the difference in Y coverage in the fetal versus the maternals in each case. So the current options in prenatal testing include amniocentesis at 15 weeks, CVS available as early as 10 weeks, and NIPT also around 10 weeks. TRIC has the advantage that you can do it as early as 5 weeks. It's also non-invasive, which is an advantage there. So in summary, TRIC uses a non-invasive cervical sampling method. It gets hundreds of cells at a very high purity, and it's effective as early as 5 weeks. It's not affected by obesity, and it provides intact fetal cells, each containing a complete human genome. It can accurately sequence the fetal genome, and it isolates extravillous trophoblast cells entering the reproductive tract, which also brings in the potential of molecular profiling that could be useful for predicting maternal risk for perinatal diseases. And finally, I'd like to acknowledge our team in my lab and Dr. Drulow's lab, as well as people in the Department of OBGYN who have helped us with samples. We're keen to find collaborators like those that we have here who can help us as we move this towards something that can be used clinically. And so thank you for your attention.